All right, let's get started. Are there any questions? So we finished 3 chapters last time, right? So I'm going to start on chapter 4 now. Right? And the title of this chapter is Water Balance of Plants. So water balance of plants. So to find out, to describe how water is taken up by plants from the soil, right? We know that, soil can be of different types. If you have sandy soil, that is not good at water retention, right? And the soil particles are much larger. So sandy soil has got much larger particles and cannot retain much water. On the other hand, if you have clay type soils, the particles are much finer and they can retain a lot of water. Now, in order to extract water from the soil, right, the plant will have to use some mechanism. Now, the basic laws of physics that I talked about, all this surface tension, water potential, right, those things will have to be obeyed, okay, while water is being extracted from the soil. Right. Now, water moves through the soil by what is called bulk flow. So, water moves through the soil by bulk flow. Right? And if you have water spread out all over the soil, right, the portion from which it will be removed first, that is going to be decided by things like water potential and all that, okay? And that is discussed in detail in that chapter. I'm not getting into that, right? Now suppose the root of a plant absorbs water, right? So plant root absorbs the water from the soil. Then in that area of the soil, the pressure potential is going to fall, right? So pressure potential falls near the root surface. Right? Which means that water from elsewhere will come in, right? Because the water is going to go from the higher potential to the lower potential. Right? So water from elsewhere will come in. Now the roots also have something that are called root hairs, right? Because if you increase the surface area, right, you're greatly going to facilitate the absorption of water. So root hairs provide additional surface area for water absorption. Now once the water is absorbed from the soil, right, by the roots, then the water moves in the root via the apoplast. Anybody remember what's the apoplast? It's basically the network that is made up by all the spaces in the cell wall, right? So that's the apoplast. And then we have the symplast, which is the network that is made up by the interior of the cells that are linked together by plasmodesmata, right? And also by transmembrane pathways. And for this last one, right, transmembrane pathways, the transport of water is facilitated by what are called aquaporins, right, which are basically like channels in the plasma membrane that allow water to go through, right? So facilitated by aquaporins. And I talked about these last time. Now today, I want to talk about how water is transported, let's say from the roots to the leaves, right? How does that happen? Now that takes place through what is called the xylem, right? So we are going to talk now about water transport through the xylem, right? And there is one thing in particular that I want to point out. So water transport through the xylem, right? So the xylem is like a system of pipes, right? It's made up of smaller units that are put together, right? And then water, and these are all dead cells, right? The cells that have developed the xylem, they are all dead cells. So it's like pipes over there, and water is going to flow through it, right? Driven by the water difference in the water potential, right? Water is going from higher potential to lower potential. Now, the thing that I want to point out that if you look at the units that make up the xylem, all right, these units are called tracheids and vessel elements, right? And it's basically a whole bunch of elements, right, that have got holes in them, right? So a typical unit might look like this. Again, excuse my diagram, it's not very good, right? But you're going to have holes here, right? You're going to have holes here, right? And then holes here also. At the same, so these are going to be connected like end to end, right? Roughly speaking, for water transport. But you also have holes on the sides, right? Small holes on the sides, right? And you have neighboring units that are linked together like this, right? So it will not just be one set of units that are longitudinally connected together, but laterally also there will be other units, right? And I want to show you what that's going to achieve, right? Because these guys, they have these things on the side that are called pits, right? This will allow the xylem to avoid cavitation, right? Remember last time I talked about that if you have an air bubble, right? It is possible that that air bubble could grow, right? And completely shut off the water supply, right? So here, because these guys are connected, say you have a big air bubble that builds here, right? Right? But there will be connections between these pits, okay? So it can bypass the one in the middle, connect with the other one, and then water is still flowing, right? So pits on the side, so that's what I wanted to point out, right? Because I talked about this cavitation thing last time in the context of discussing surface tension, right? So pits on the sides, they help in avoiding cavitation in the xylem. Because if that happens, right, if there is cavitation, then the plant is not going to get any water, right? At least a portion of the plant, that part is going to die. Okay. Now, one thing to note is that, in the xylem, all right, as I said, this is like a network of pipes, all right? So it basically provides a pathway of low resistivity for water flow, right? It is much easier, right, to make water flow through the xylem because this is just like a pipe as opposed to trying to drive water through cells because then you have to cross the cell membrane, right? Then there is resistance inside, right? There's the cytosol and so on, okay? So the xylem provides a pathway of low resistivity for water flow. And also the cohesion tension theory. Right, remember I talked about the capillary tube and all that stuff, right, last time? So that's the cohesion tension theory because you have surface tension and then you're talking about cohesion, then the contact angle being small and so on, right? So the cohesion tension theory explains water transport in the xylem. Right? And water is going to be drawn up from the roots, right? all the way up the shoots to the leaves. Okay, what's going to happen to the water once it reaches the leaves? It's going to just evaporate. There's going to be transpiration, right? So what is the driving force for that transpiration? It's the difference in vapor pressure, right? The water vapor pressure inside the leaf versus the external environment, right? So if it's dry weather, there will be more transpiration, right? So the driving force for transpiration is the difference in water vapor pressure or water vapor concentration, right, between the leaf air spaces and the external bulk air. And the diffusional resistance of this pathway is also a factor, right, in deciding how much of transpiration takes place. So the diffusional resistance of this pathway is also a factor. Now, the leaves have openings in them that are called stomata, right? That can open or close to allow either more water to come out, right, or less water, right? So leaves have openings called stomata. So leaf stomata can open or close as needed. So if conditions are such that too much of water is going to be lost, the leaf stomata should close, right? And how is that achieved, right? That is achieved by a change of turgor pressure in the stomatal guard cell. So you have this opening called a stomata. Around it you have two cells that are called guard cells, okay? If there's a lot of water inside those cells, right, the turgor pressure will go up, right? That is going to cause the stomata to open, right? So if turgor pressure in stomatal guard cells increases, then the stomata will open. Stomata will open, and vice versa. And I will discuss mechanisms that will cause that turgor pressure to increase, right, when I talk about photosynthesis and other things. Right. All right, any questions? So for this particular chapter, we had already laid out the groundwork, right, last time. So this is really a pretty short chapter. So we've just discussed a little bit. I wanted to point out the important things. And any questions on this chapter? If not, we will move on to the next chapter, right, which is chapter 5, which is on mineral nutrition. And again, I'm summarizing all these chapters, picking up the important points, right? If you have the time, or if you're going to work in one of these areas, you can get hold of the book, read the chapter, and you need to, because you're not going to be working on the topics highlighted in each and every chapter. Whichever chapter you focus on, you spend the time to read it. You might have to read do some literature survey and so on. Okay. But I'm trying to give you an overview so that the first time you look at something, you're not completely lost. You have the big picture. So this chapter is about mineral nutrition, right? So in general, most plants are what are called autotrophic organisms, right? That means they can produce everything that they need, right? So can synthesize all components from the naturally available things like carbon dioxide, right, water, and minerals, right? So the plants will get carbon dioxide from the air, right? But the water and minerals will come from the soil, right? So water and minerals come from the soil. So there are many mineral nutrients that are essential for plant life, right? So there are specific mineral nutrients essential for plant life, right? And I'm just listing a few of them. For example, nitrogen, right? Phosphorus, potassium, and so on, right? Why is nitrogen essential? What are you going to need nitrogen, right? For making what? What kind of molecules in the case of a living organism are you going to, Go ahead. What? Exactly, right? For making DNA, RNA, even for proteins, those are amino acids. So you're going to need nitrogen, right? Phosphorus, again, I mean, you'll need it for many different things, but one of the things that you'll need it for are for what? Well, yeah, kinase you will need, and even in the case of DNA also, okay? Like you have those nucleoside, there's a sugar phosphate backbone, so you're going to need phosphorus, right? So things are falling in place. This is not just something pulled out of thin air, right? Now the nutrients that a plant needs are usually classified as either macronutrients or micronutrients depending on how much is needed, right? So classification as macro and micro nutrients depending on the relative amounts found in plant tissues. And if you don't have enough of these mineral nutrients, right, there's going to be severe deficiencies. The leaves might turn brown, yellow, and things like that, right? They might fall off, right? So you'll see those symptoms because these nutrients are needed in several of those life processes, right? So nutritional deficiencies can lead to various symptoms as minerals serve as components of organic compounds in energy storage. Remember ATP, right? You need nitrogen, you need phosphorus, so energy storage. Then enzyme cofactors and so on. And even enzymes are proteins, so you will need nitrogen, right? So you can test the soil and plant tissue analysis, right? You can analyze the soil, you can analyze the plant tissue, you can determine deficiencies, take corrective action, right? So soil and plant tissue analysis indicates nutritional status of that particular plant and suggests corrective actions. Now for growing plants, right, you really don't need soil. If you supply the roots of the plant with the appropriate minerals, right, and water, right, you will not need soil, okay. Soil is something that is just, fixing the plant to the ground, right? So typically in the lab, they do what is called a solution culture to study plants, right? So solution culture to study mineral nutrition. So solution culture to study mineral nutrition. And there are several options for doing that, right? So you have to make sure, right, that when you don't have the roots embedded in the soil, but you're going to give the roots enough water, right, and nutrition. And you also have to make sure that the roots get enough oxygen, right? So you must make sure, must make sure that roots get both minerals, right, and water and oxygen. And there are a number of possibilities for doing that, right? You could have what is called hydroponic growth system, right? Right? So in the case of the hydroponic growth system, right, you have a nutrient solution, the roots are immersed in that, okay? But you are also blowing air bubbles to make sure that the plant is getting oxygen. So nutrient solution with air bubbles. And if you want to see pictures and all, you can look at that in the book, all right? So nutrient solution with air bubbles. Number two, you could have a situation where there is a thin film of nutrients, right, to facilitate the growth, right? So nutrient film growth system. Then another possibility, right, would be to use an aeroponic growth system where the roots are actually suspended in air, but you're going to periodically spray them with the nutrient solution, right? So aeroponic growth system. So you spray the roots intermittently. And the last method that is used is what is called the ebb and flow system, where you have the water, where you have the solution rising up, immersing the roots, right, then coming down. Okay, so the roots get air. So ebb and flow system. So if you interact with some plant biologists, you will see they are using one of those systems to grow those test plants in the lab. Any questions? Now, it is a fact that you can add fertilizers to the soil, right, to improve the plant yield. Okay, now why does that happen? So the addition of fertilizers can improve the above ground growth and yield, right? And why is that? That's because, see, the roots have to go searching for water and for nutrients in the soil. Okay, so if you don't add fertilizers, the roots will have to go deeper and deeper. There has to be growth over there, right? If you add fertilizers, roots don't need to grow. They're getting the stuff right away. So that excess growth can take place above ground, right? So you can have vegetative and what is called reproductive growth, right? That is enhanced, right? So that's the principle behind fertilization of plants to increase the crop yields, right? Another thing is that plants can get carbon dioxide from the air, right? Oxygen also from the air, water from the soil, right? But the plants also need nitrogen, right? And nitrogen is available in very high concentrations in the air, right? I mean, it's like 80% almost, right? But the plants cannot go and capture that nitrogen, right? That reaction to capture the nitrogen from the air is like energetically unfavorable. They cannot do that. They have to take it from the soil, okay? And several bacteria have, on the other hand, have the ability to absorb nitrogen and process it to a form where the plants can absorb that, right? But those bacteria need food in order to survive, right? So the plants can provide them with the carbohydrates because through photosynthesis the plant can produce carbohydrates, right? So there can be a symbiotic relationship between bacteria and plants, right? So many plants, have symbiotic relationship with nitrogen fixing bacteria these bacteria supply nitrogen to the roots while receiving sugars. So while receiving sugars from the plant. And in addition to bacteria, there are also what are called fungi that are called mycorrhizal fungi, right? That do the same kind of thing. So also my CORRHIZAL fungi. Now, if the plant absorbs excess minerals from the soil, right, that can also be toxic for the plant, right? So there has to be some balance. So excess minerals can be toxic, and the absorption of minerals from the soil, that could depend on the pH of the soil, right? The plants have the ability to alter the pH, right, because they can shoot out hydrogen ions, right, into the soil. If the hydrogen ion concentration goes up, the soil will become more acidic, right? So soil pH or hydrogen ion concentration, H plus ion concentration, affects nutrient availability. Because depending on the pH, the plant may be able to absorb a nutrient or not. Okay, nutrient availability. Then the soil microbes also might need an acidic environment to survive, and even root growth will depend on the acidity of the soil. Right? And we will talk more about how hydrogen ions are pumped out to control the acidity. Okay, later on. Right? Now the last thing to note in this chapter is that as the plant is absorbing these nutrients from the soil, right? So the region immediately surrounding the roots will be depleted of nutrients, right? So you have to keep supplying that region, right? If you fertilize somewhere further away from the roots, then the roots need to grow over there, right? So that they can absorb the nutrients. So plant roots continually deplete the nutrients from the immediate soil around them. All right, any questions? Right. So that finishes this chapter on mineral nutrition. Again, it's just a survey, right? Important things that caught my attention, I'm trying to highlight that, and things which are easily explained to engineers, right? You cannot become, like, you can probably focus on one chapter and do research work in that area. That's a different thing, okay? But you cannot become a plant biologist, okay? That's not what we're trying to become, right? So the important things, at least to me, what seem important, I'm highlighting those. We move on to, so no questions so far. Okay. So we move on to the next chapter, which is on solute transport. And again, the focus will be on things which can be easily explained to you, right? Now, solute transport also is going to be driven by the chemical potential, right? I mean, because for any reaction, it has to take place in a direction where the free energy goes down. Okay, we know that from the second law of thermodynamics. Okay, so whether the solute is transported or not in a particular direction is going to depend on the difference in free energy or chemical potential. So here, see, we might also be dealing with charged molecules, right, that have charges. So, it's not just the concentration, it's also the charges that will determine. Concentration and charges together will determine the direction of flow, right? So we'll try to capture that. So to capture that, suppose you're looking at the inside and the outside of, let's say, of a cell, right? Or you're looking at two compartments that are separated by a membrane, right? So let's say you have, this is a membrane here, and you're looking at the electrochemical potential outside and inside the membrane, right? So anything that pertains to stuff inside the membrane, I will denote it by this superscript I, right? Anything that is outside, I will denote it by the superscript O, right? And let's say for the time being, for the sake of simplicity, we are focusing on one single solute, right? Let's call that solute J. In general, inside the cell, there will be many things, okay? So first, let's focus on one of them, right? So suppose we have this should be O. Outside means O. So suppose we are focusing attention on solute J, right, with different concentrations inside and outside, right? And also the voltages inside and outside are different, right? So electric potential. So inside and outside. And the inside and outside, as I said before, are separated by a membrane. So inside and outside separated by a membrane. So now if the solute also has electric charges associated with it, so if solute has electric charges associated with it, we need to consider what is called the electrochemical potential. So it's going to take the chemical potential, but in addition to that, even the effect of electricity has to be taken into account, the charge differences. So we need to consider the electrochemical potential both inside and outside this cell. So for the electrochemical potential inside and outside, we can write down two equations because each of these guys will be having different components. We are going to sum them together. So for the electropotential of solute number J inside the cell, we will have this kind of relationship. Mu tilde Ji, right? The electrochemical potential is going to be the sum of mu J star, right? So this is the potential under standard conditions, okay, when the other terms are not there. So this is the potential for the solute under standard conditions. That means when all the other terms drop out. So potential under standard conditions. Then plus, there will be a term because of the solute concentration inside, right? And that is given by RT times the natural log of the concentration of the Jth solute inside the cell, right? And so this is the concentration term, right? Then there will be another term because of the electric charge on the solute, right? And that is given by Zj, right? Let's say the solute, if it happens to be like the sodium ion, okay, it's +1, so Zj will be +1. If it's a chloride ion, the charge on that is minus 1, so Zj will be minus 1, right? Times capital F, which is the Faraday constant, right? And times the potential the electric potential inside the cell, right? And then there will be another term because of the pressure. So this is the term due to charges. Term due to electric charges, right? And then plus V bar J times Pi. This is the term due to the hydrostatic pressure. Right? So now we can also write down a similar equation, right, for the electrochemical potential outside the cell, right? Because we really want to see if something is going to flow through the membrane or not, right? So, or how do we regulate that flow? So Mj tilde outside, it will be the sum of the same term mu j star, okay, because this is the potential under standard conditions, right? Mu j star plus RT ln Cj, it's going to be Cj what? O, yeah, outside, plus Zj F EO, right? Plus the next term, V bar J PO, right? And let me just make a note, because in case you decide to verify the calculations that I'm going to mention, this R is the universal gas constant, and the value for that is 8.32. So universal gas constant, and the value is 8.32, right, whatever the units are. And then F is Faraday's constant, and the value is, so F is Faraday's constant, and the value of that is 96,500 coulombs. So now to figure out, what flow is going to take place across the membrane, we have to take the difference between these two potentials, right? Now usually that pressure term, that last term over there, the contribution from the last term is usually small, unless you're looking at, osmosis across the membrane, right? That last term will drop out, okay, or we won't consider that. And so the contribution from the last term is usually small, right? So we will ignore the difference in the last term for these two equations, right? And if we take the difference, right, then we will get the delta mu tilde, which is mu j tilde I, right, or delta mu tilde j, right, is mu j tilde I minus mu j tilde O. Okay, that's giving you the electrochemical potential difference across the membrane. That is going to be equal to what? The mu j star terms will cancel out, okay, because that's under the standard condition. So you will get, this is going to become RT natural log of Cji, right, divided by Cjo, agree, right? And then the next term is going to be plus Zjf times Ei minus Eo, right? Okay. And this is actually the voltage difference across the membrane, right? So this is, we can call this delta E, right? Now see, if delta mu j tilde, right, if this quantity is less than 0, right, then what can you say? If this is less than 0, that means the potential, electrochemical potential outside is more or less? More outside. So that means the flow should be in which direction? The spontaneous flow? Outside to inside, okay. So implies potential outside, potential outside is higher, which means that the spontaneous flow is inward. Okay, this makes sense to you, right? I mean, if the concentration, okay. Now, all right, I mean, for a moment, if we forget about the charge, okay, let's say it has no charge, right? Yeah, I'm saying the potential is higher outside, therefore the flow will be inward. But let's say we forget about the charge. The last term, let's say the molecule is uncharged, okay? Then the flow should be determined by the concentration gradient, right? So in that case, if the concentration outside, right, Cj0 is higher than Cji, all right, then RT ln Cji over Cj0 is going to be negative, right? I mean, because it'll be something log of something less than 1, and then this agrees with whatever we predicted based on this equation, right? So this makes sense. So this makes sense if Zj equal to 0, that means no charge on the solute that you're focusing on, and the concentration outside happens to be higher than the concentration inside, right? Then flow should be driven by the concentration gradient. Then flow should be inward. And that's what the earlier equation tells us. Okay. Now suppose we have actually a charged molecule, right? We have a charged molecule. And let's say the concentration outside is much higher than the concentration inside. But we don't want any flow to take place. Okay. How can we do that? We can do that by applying a voltage. Right? So suppose we want no flow despite concentration gradient. So if you don't want any flow, the difference in that electrochemical potential has got to be 0, right, across the membrane. So implies you can apply, right, by applying an electric field you can make that potential difference between the inside and outside, the electrochemical potential difference 0. So apply electric field. So what is the size of the electric field that you're going to need to apply, okay, or how much voltage do you have to apply? Well, basically these two terms here, right, here, they have to sum up to 0, right? Then there'll be no flow from outside to inside, no flow of the solute, all right? So if you write down, if you set that equal to 0, you will get the equation RT ln Cji divided by Cjo plus Zj F times delta E equal to 0, which means that delta E must be equal to RT over Zj F natural log of Cjo over Cji, and this quantity that you calculate, right, so there's no flow, this is called the Nernst potential, right, of that solute. And to get a better feel for this equation, right, so suppose, let's take a more concrete example. Suppose you have a positively charged ion, right, the concentration outside is 10 times higher than the concentration inside. So if ion is unit positively charged, that means the charge is +1, like it could be sodium, potassium and so on. So unit positively charged, so Zj equal to 1, right? And let's say the concentration of this ion outside is 10 times the concentration inside, right? Cji equals to 10. Then you do the math. Okay, I gave you the values for all this, Faraday's constant, universal gas constant and so on. Then you calculate, you get delta E equals 8.32 times 298, right? Because the temperature, let's say, is 25 degrees Celsius, which in degrees Kelvin is 298, right? It's 298 times the natural log of 10, right? Divided by 1 times 96,500. You calculate and you will get 0.059 volts, right? Or 15 millivolts, right? So if the voltage inside is 15 millivolts higher than the voltage outside, despite having this big concentration difference, right, the solute is not going to flow. So if So if EI, the voltage inside, happens to be the outside voltage plus 0.059 volts, right? No solute movement will take place. And if you actually make the voltage larger, right, the difference larger, then actually solute will flow out, okay, from inside to outside, okay. All right, any questions so far? This is stuff close to double E, so that's why I'm trying to, not make it just a complete plant biology class. Any questions? All right. Now, see, what we talked about here is the Nernst potential, right? Or the Nernst equation. Now, inside the cell, you'll actually have many different charged particles, all right? So you have to combine the effect of all those particles, all right? And that is given by what is called the Goldman equation, and you talk about the Goldman diffusion potential, all right? So, and the potential is basically that voltage. So Goldman equation and Goldman diffusion potential, that actually combines effect of different kinds of solutes. And that's of course going to be a much more massive calculation, and we are not even going to get into that. Now we look, so now we know how you can generate an electrochemical potential difference across a membrane, right? So you can use that to drive transport, right? So if the thing flows down the electrochemical potential difference, that is called passive transport. No extra energy is needed, right? Like water always flows from higher potential to lower potential. Electricity flows from high, so the same thing will happen here, right? So passive transport is basically transport down the electrochemical gradient. Right? And active transport will be just the opposite, right? You can go uphill, but you have to supply energy, right? If you want to move water up, you have to supply extra energy. So active transport is transport against the electrochemical potential gradient. So it's uphill transport. And that requires energy input. And for a cell, where is that going to come from? ATP, yeah. Right? So you will have some, and they're called pumps, okay, because they'll push things uphill, right? They'll use up ATP. So they're called ATPases because they basically degrade ATP and get the energy from there, right? And there are two examples of that which we will probably encounter many times, right? And I just want to mention that here. So if you look at a cell membrane, right? So let's say you have a typical plant cell here, and then inside you have a vacuole that has a membrane, right? So this is the plasma membrane. And inside the cell, there are different organelles. I'm just picking these two, right? So this is a vacuole, right? What is the membrane of the vacuole called? Anybody? Tonoplast. Right? I mentioned that last time, right? So here what you can have is the following situation, all right? And this happens quite frequently, all right? So you could have an active pump, right? That pumps out hydrogen ions from the plasma membrane outside, right? So hydrogen ions are pumped outside, right? And you need energy for doing that, right? Because see, if you pump some hydrogen ions outside, the hydrogen ion concentration outside will increase, right? And then you have to still keep on pumping. So you're doing it against the concentration gradient, right? Or the electrochemical gradient, right? So protons or hydrogen ions, so protons pumped out of cell by what is called a proton pump or a hydrogen ion ATPase. This is an example of active transport because ATPase has to use the energy from ATP, from the hydrolysis of ATP to pump the hydrogen ions out, right? And the cells have to do that because the outside is usually more acidic. Hydrogen ion concentration is higher. Okay. Same way in the case of the vacuoles, all right? There are some ATPases that are going to pump hydrogen ions into the vacuole, right? So vacuole. So you have vacuolar hydrogen ion ATPase pumps hydrogen ions into vacuole, right? So this is the mechanism. Like if you look at lemons and lime, these are sour, right? So there are hydrogen ions inside the vacuole, right? That's what's giving it its sour taste. If you look at the stomach of an animal, right? There's hydrochloric acid in there. How is that produced? Well, the cells lining the stomach, they are pumping protons in, right? And if they pump too many protons in, then you have problems. Okay, you have like stomach acids and acid reflux and blah blah blah, right? And people suffer from that. So this is close to home, right? I mean, it's not some esoteric stuff that I'm talking about, right? So these are proton pumps, right? So passive transport is basically transport down the electrochemical gradient, right? So in this case, suppose suppose you had this ATPase, all right? It has made the hydrogen ion concentration much higher outside. So the natural tendency of the hydrogen ions would be to move inside, right? So that's like a downhill transport. Now, if you can couple that thing, right? If you can couple the movement of hydrogen ions down that hill with energy production, with ATP production, then you're doing exactly the same thing that we do to generate electricity from water power, right? You pump water up the hill, then you let it come down, and you basically use that to turn your uh, generator, right? Produce electricity. The cells do that all the time, right? By the time we get done with all this stuff, you will see it's done in photosynthesis. It is also done in respiration, both in plant as well as in animal cells. So we'll talk about that. Right? So passive transport is made possible by channel proteins or carrier proteins, right? So made possible by channel proteins. So you could have a small channel, right, through which some molecule could flow down the electrochemical gradient, right? So and those channels are called channel proteins. So no energy is no extra energy is needed. You're going down the electrochemical gradient, right? Or you could even have what are called carrier proteins, all right? So carrier proteins, these are these might be things, you know, there might be things on the membrane, right? That the thing is that some molecule is flowing through, right? And you basically hitch a ride with that molecule, right? So it's like piggybacking, right? So carrier proteins facilitate that, but again, no external input of energy is needed. Some molecule is going through because of the electrochemical gradient, right? It's passing from one side of the membrane to the other, and you just hitched a ride with them, right? And there are many different carrier proteins that are there. We don't have time to discuss all of them. So this is passive transport, right? Now, if you have active transport, again, any questions? You can have two types of active transport. You can have primary active transport, and you can also have secondary active transport. Again, remember, active transport is going to be against the electrochemical gradient, all right? So it is going to require an input of energy. In the case of primary active transport, you will basically be using pumps, all right? So this uses pumps. An example of that I already gave you, the hydrogen plus ATPase, right? And there are other pumps too, like sodium potassium pump and so on, okay, in the cell, right? To keep the concentration of sodium and potassium in the cell at certain levels, right? If you're looking at secondary active transport, right? So normally, right here, maybe some molecule is not going to flow in a particular direction, right? But hey, if you can couple this reaction with something else, right, that is going to happen, right? That is happening anyway, right? Then you're in good shape, right? So secondary active transport will uses stored energy. Example, hydrogen ion entering the cell. So hydrogen ion entering cell. So you pump the hydrogen ions outside, okay? So the hydrogen ion can now enter the cell, right? Because of the gradient, right? The proton gradient that has been established across the membrane. But in the process, it can carry something else along with it, right? Either inward or outward, right? So example, hydrogen ion entering cell down the electrochemical gradient can carry some other ion against its electrochemical gradient. So if both of them are traveling in the same direction, they're crossing from one side of the membrane to the other, both of them are headed in the same direction, then that is called symport transport, right? So if both in same direction, then this is called symport transport. And if the other ion, right, moves in the opposite direction, then this is called antiport transport. Now the pumping of hydrogen ions from inside the cell to outside, that produces what is called a proton motive force across the membrane, right? Because you have a higher concentration of hydrogen ions outside, lower concentration inside. So there is a tendency for the protons to move inside, right? And as I said before, it can be used to generate ATP. So the proton motive force produced by hydrogen ion pumping can be used to generate ATP, right? And we will see examples of that in the chapter on photosynthesis, right? And also when I talk about respiration later, okay, in the mitochondria. So this is used in what is called the electron transport chain in mitochondria. Also, you'll see that during photosynthesis in chloroplasts. Also, during photosynthesis in chloroplasts. All right, any questions? So we finished this chapter too, right? And that was solute transport. So now I'm ready to move on to the next chapter, that is chapter 7. And this chapter is about photosynthesis. And it's photosynthesis is made up of two parts. Right? There are some reactions that are called the light reactions, the one reaction involving light, and then there are other reactions that are called the carbon fixation reactions. So chapter 7 is photosynthesis, the light reactions. Right? Now let's start off by taking a look at the overall reaction of photosynthesis. So the overall reaction of photosynthesis goes as follows. You have carbon dioxide plus water, right, in the presence of sunlight. It gives you CH2O, right? That's a carbohydrate, carbon plus water, right? So if you have 6 molecules on either side, you will get glucose, right? So CH2O plus oxygen. Oxygen is released, right? And this is a carbohydrate. Now this reaction can be divided into two parts. The first one, part 1, these are called the light reactions. Also called the thylakoid reactions. And I will explain the reasoning behind this name thylakoid reactions in a few minutes, right? And what happens in this set of reactions is that you have two molecules of water, right, in the presence of sunlight. They give you 4 hydrogen ions, right, plus a molecule of oxygen and 4 electrons, right? Now the energy over here that is released, right, in the form of these electrons, that is actually captured in energy carriers. And what are the energy carriers that are possible? Based on what we have already learned in this course, what are the possibilities? Which are the molecules that can carry energy? Yeah, go ahead. And? And which molecule can carry high energy electrons? NADH and NADPH. Okay, NADH is for the metabolic reactions, right? I'm sorry for the break catabolic reactions. That is like respiration and all that. Here these will be biosynthetic reactions, so it is NADPH, right? So so captured in energy carriers ATP and NADPH, right? So this fellow, this ATP carries energy in its high energy phosphoanhydride bonds. NADPH carries energy in the high energy electrons. Okay, provides reducing power. So provides high reducing power. Now the part 2, those are the so part 2 of photosynthesis is where we have the carbon fixation reactions. Okay, so you actually end up getting these carbohydrates, right? So these are what are called the carbon fixation reactions. So carbon dioxide from the air is produced to give you carbohydrate, and these are also called the stromal reactions. Right? And basically in these stromal reactions or the carbon fixation reactions, the ATP and NADPH that has been produced in part 1, that is going to be used to drive the second set of reactions. Right? So ATP and NADPH from part 1 are used to drive carbon fixation. So the carbon fixation reactions will be discussed in the next chapter. So in this chapter we focus on the light reactions. Okay, so now I would like to explain to you, right, why these light reactions are called the thylakoid reactions and the carbon fixation reactions are called the stromal reactions, right? So if you look at a chloroplast, right, a chloroplast, as I said before, has got how many membranes? Two membranes, okay? So if you look at a typical chloroplast, you're going to have two membranes. And again, excuse my drawing because I'm not a good artist, but I think I can still explain. And if you want to see better pictures, look in the book, right? So you'll have so one lipid bilayer, then there is another lipid bilayer in the chloroplast, right? Right? Then in addition to that, right, inside the chloroplast, you're going to have some stacked membranes, right? So those membranes, they are called thylakoid membranes, right? So you're going to have some membranes that are stacked inside, right? One on top of the other, like this, right? So these are called the thylakoid membranes. Right? And this stack over here, so you'll have a number of them inside the chloroplast, many of them, right? Stacked membranes. This stack over here has a name. It's called the grana lamella, right? So G-R-A-N-A, grana lamella. Okay. Now, so you have units like this inside the chloroplast, like stacked membranes, right? These are connected together by some unstacked membranes, right? So just one stack in it, right? Or one layer in it. Those are called, so it's coming out, let's say from the bottom here, right? That part is called the stroma lamella, right? So you will have many of these grana lamella that are connected by stroma lamella. So let's put in that terminology here, right? So you can see what the thylakoid is. The thylakoid membranes, right? So these are the membranes that are stacked one on top of each other. So the light reactions actually take place in the thylakoid membranes, right? That's why they are called thylakoid reactions, right? The carbon fixation reactions actually take place in the interior of the chloroplast. Whatever is not the thylakoid, right, or the stroma lamella, that is the stroma, right? So this region of the chloroplast is called the stroma. And the carbon fixation takes place in the stroma, so they are called the stromal reactions. Okay. Now, one more thing that I want to point out is that even in the case of the light reactions, right, this takes place there are two photosystems that are involved that work in series. Photosystem 2, and I will refer to this in a few minutes, all right. PS2 is photosystem 2, right. I will write out the full form later. That takes place in the grana lamella, right. While the other reaction, which is called PS1, photosystem 1 reaction, that will take place in the stroma lamella, right? So just keep this in mind. Since I've drawn the figure, I'm just inserting all these things right now, but I will need to refer to these. Okay. So first the reaction takes place in the PS2 reaction takes place, the photosystem 2 reaction will take place. That is followed by the PS1 reaction. And notice the locations are different. One of them is taking place in the grana lamella, right, which is the thylakoid membrane stacked up. The other one is taking place in the stroma lamella. So if the first reaction is going to feed the second one, there has to be some transportation also going from one place to the other, right? So keep these things in mind, right? So stroma lamella is the site of, the stroma, this one is the site of carbon fixation. All right, any questions? Any questions? So from the last picture, we can summarize that the light reactions occur in thylakoid membrane, and the carbon fixation reactions they occur in the stroma. And that explains the nomenclature for these two types of reactions. Okay, so now before we can discuss the light reactions, let's just recapitulate a few facts that light is a form of energy, right? And this is stuff that you as engineers you should be knowing, right? So light is a form of energy, right? Light behaves both as a particle as well as an electromagnetic wave, right? Behaves both as a particle as well as an electromagnetic wave. And the energy in light, in a photon of light, is given by the expression E equals h nu, right? Where h is Planck's constant and nu is the, what is nu? Frequency, yeah. So nothing new here. And then also we know that we can relate the frequency to the wavelength of light just by c, the velocity of light equals lambda times nu, where lambda is the wavelength. Now if you look at visible light, what is the wavelength of visible light? What is the wavelength range? Anybody remember? Or? The wavelength of visible light goes from 400 nanometers to 700 nanometers, right? So 400 nanometers is basically the wavelength associated with violet light, right? And 700 nanometers is the wavelength associated with red light. That is the visible spectrum, right? So if you're looking at ultraviolet or infrared light, right, then ultraviolet means it will have more energy than violet, right? So the wavelength will be smaller, right? So ultraviolet has energy above violet, which means that the associated wavelength will be less than 400 nanometers, right? Because the frequency will be higher. All right, and similarly, infrared has energy below red, right? Which means that the wavelength is going to be larger, so lambda is going to be greater than 700 nanometers. Now, if you look at chlorophyll, right, light absorption by chlorophyll, that's the green molecule that pigment that you find inside chloroplast. It has got two peaks in its absorption spectra, right? So chlorophyll absorption spectra has two peaks. One of them is at 430 nanometers, which is actually blue, right? It's close to blue. We said like 400, yeah. So 430 nanometers is blue, and it has another absorption peak at 660 nanometers, right? But the green part of the spectrum, it doesn't absorb, right? That's what it reflects it to our eyes. That's why we see the color as green, okay? So the green color, this explains the green color of chlorophyll, right? Because that green part is not absorbed. Now suppose, like initially chlorophyll is in the ground state, but then it absorbs blue light, right? So let's say chlorophyll absorbs blue light. So then it will go to a much higher energy state, right? So it's in a higher excited state. Then from there, it can lose some amount of heat energy and go to a little bit lower excited state, because this higher excited state where it goes to from blue light is extremely unstable, right? So it can lose some energy in the form of heat and drop to a lower state which is still excited, lower excited state. From here, right, this chlorophyll molecule can do a number of things, right? There are a number of possibilities. There are 4 possibilities, and I will list those, and then we'll conclude the thing for today, all right? So there are 4 possibilities, all right? First, it can emit a higher wavelength photon. Why higher wavelength? Why higher wavelength than what it absorbed? Because some of the energy has been lost in the form of heat, right? The absorbed blue light, it went down, so some of the energy has been lost, so it's going to be a lower energy photon, so a little bit larger wavelength. Or, right, it could do the following, it could give out heat. Like all the energy is lost and it returns to the ground state, right? That means nothing has been achieved, right? Or the third one is that it could pass its energy onto some carriers, energy carriers, right? Or it could participate in energy transfer. And the last thing that it could do is participate in photochemistry. That is the light reactions of photosynthesis. Participate. Now it turns out that this last step here, among these 4 possibilities, the last one is the most rapid, right? And it takes precedence over everything else, right? The other things do not happen. So this is the most rapid and takes precedence over everything else. So that's the one that will happen, right? And we will start talking about the light reactions of photosynthesis next time. Just keep in mind that picture where I talked about PS1, PS2, that's going to be needed, right? Because for next time's discussion. But today, let's stop here, right?